Today we are going to talk about bacteria. On the top right side of your bacteria notes, you will find the area that we're going to start writing these notes. The first thing is that bacteria are prokaryotic, and we know that prokaryotic cells have no nucleus. So bacteria do not have a nucleus. They are all single-celled, meaning one single cell is the entire organism. This organism over here that you see is the bacterium. That's one single organism of a bacterium. This happens to be E. coli, which can cause like digestive issues. But bacteria are single-celled organisms. And because they are a single cell, they're really, really small organisms. And therefore we measure them in micrometers. Bacteria typically come in one of three shapes, and that's all we're going to need to know. There are some exceptions to this, but the three that we will need to know are the same picture that you see on your sheet. The first one on the left is the sphere shape. So these are bacteria that come in a sphere-like shape, and we call those coccus. Any bacteria that you see with the name coccus, like Staphylococcus aureus or like a staph infection, that tells you that it's a spherical bacterium. So if you look under a microscope and you see a non-sphere bacteria, you know it can't be staph because it's not that sphere shape. Now, not all bacteria have the shape in the name, but it is important to know those shapes because that's one key identifying feature of them. The middle you can see are rod-shaped, and those we call bacillus. And then the left, or on the far right, we see the spirally shape is called sporulum. Another method that we use to identify bacteria is Gram staining, and this involves staining the bacteria with a stain and seeing if that sticks or not, and if it sticks or not, it will have either the purple or the pink color. And so if you look at the third bullet point, gram-positive bacteria have a thick layer of peptidoglycan and no outer membrane. They do still have a cell membrane, but it's just not on the outside. And these bacteria are stained purple. They'll retain a purple color, and we call those gram-positive. Gram-negatives have a thin layer of peptidoglycan and an outer membrane, and these are stained pink. So this is just one other way that scientists and doctors can help identify what type of bacteria you have. We next want to look at the different parts of the bacterial cell. And so we're going to jump back up to the first two bullet points really quick, and then we'll go back down to the bottom. DNA in a bacterial cell, because they don't have a nucleus, the DNA is not in a nucleus. Instead, it's just clustered inside the cytoplasm. So there's nothing surrounding the DNA, but we call that clump of DNA, we call it the nucleoid. In bacteria, DNA is clustered in the nucleoid. And then they have a rigid cell wall. So animal cells don't have a cell wall. Plant cells have a cell wall, but it's made of cellulose. And in bacteria, they have a cell wall. It's made of peptidoglycan. So I know that's kind of a weird, funky word, but it is important that you understand that bacteria have a cell wall. That's a key characteristic of them. Then if you jump below, you'll see the same exact picture on your notes. I would like you to go ahead and label those different parts of the bacteria cell. If you flip your paper over, you'll see it says bacteria can be both. Number 1 is autotrophic, and then if you jump down to number 2, the second one is heterotrophic. We know this from last semester that autotrophs are going to make their own food. Some bacteria can make their own food, but some bacteria can't, and they must consume other organisms, and those are heterotrophic. The bold piece, autotrophic, make their own food. Heterotrophic cannot make their own food. Those are the two definitions that you should write after each word. In autotrophs, they can be chemo or photoautotrophic. If it's chemoautotrophic, they're getting their energy from inorganic sources. These would be bacteria that are in the depths of the ocean where there isn't any light. But photoautotrophs are going to get their energy from sunlight, just like plants. And then heterotrophs, those have to either consume other organisms by eating them and absorbing nutrients from dead organisms, or they're parasitic. Bacteria are pretty hardy and they're able to survive harsh conditions. One way that they do this is by forming endospores, and these are little kind of capsules that help them to resist harsh conditions in their environment and then they can actually come out of that state of hibernation essentially and be able to survive for long periods of time. So that's one unique adaptation that bacteria have. For reproduction, because they are a prokaryote, they have to reproduce asexually. There's no sexual reproduction with bacteria, and they do so through binary fission. Remember, it looks very close to mitosis, where one cell becomes two new cells, but we cannot call it mitosis because it's not the division of the nucleus. There is no nucleus. So instead it's called binary fission. Binary meaning two cells are made. Fission is the separation of those. So the splitting of those cells is asexual reproduction in bacteria. One thing that is really cool, but is also a very big challenge to scientists when it comes to bacteria, is that bacteria have the ability to exchange DNA with each other. This would be, for example, if I walked up to another human being and I put my hand on their arm and was able to take on their DNA and incorporate it into my own DNA. We can't do that as humans. That would be really, really cool and kind of crazy. But instead, bacteria can. Bacteria can exchange DNA. They can take some other bacteria DNA on. They can give their DNA to other bacteria. And this can happen through one of three ways. Bacterial transformation is the first one. And that's where a bacterial cell just loses some DNA into the environment, whether that cell died or something, and another bacterial cell comes and picks it up. That's transformation. So it's transforming it from one bacterium to another, and it's incorporating it into its own cells, as you can see at the top in A. B is bacterial transduction. Transduction happens when a virus takes a part of DNA from one bacterium and then turns around and it goes to the new bacteria and it injects some of that bacterial DNA into it. So it's still getting other bacterial DNA, but it's using the virus kind of as that vector, the means of transferring it over. And then the third thing is bacterial conjugation. And this is where you can see the bacteria are actually connected. They connect through a tiny little kind of straw-like thing, and the plasmid, which is a little ring of DNA, is transferred over. And so it's going to be moved over through conjugation. And that would be like the example where I said if I put my hand on somebody's arm and gave them some of my DNA, that would be kind of like bacterial conjugation. The reason this is a challenge is because it allows bacteria to evolve rapidly when it comes to resisting antibiotics and antibacterial soaps and things like that. So these mechanisms are highly efficient and beneficial for the bacteria, but they present a really large challenge for us as humans trying to fight off the more deadly, harmful ones. The last slide here is just some diseases. I would like you to take a look at these, read about the pathogen that causes the disease, the area that is affected, and the mode of transmission, because we're going to be doing a project. My regular kids are going to be doing a project on a disease of their choice. And so I want you to start kind of thinking about what type of system do you want to look into that gets affected by a bacterium or a virus. But these are some bacteria that you could choose from.